Today we will study histology of male reproductive system. Male reproductive system comprises of main reproductive gland, that is testis. There are few genital ducts. There are epididymis, vas deferens and ejaculatory duct. Accessory reproductive gland, that is prostate, seminal vesicle and bulbourethral gland. And lastly, organ of copulation, that is the penis. Now coming to testis, which is the main reproductive gland of male. It produces sperm cell and testosterone. Its function, which is very much important. Examiner always used to listen these words, that what is its function? It produces sperm cell and testosterone. Another one is the genital duct, that is epididymis. Here it is. This is epididymis. This is head of epididymis. This is body and this is tail of epididymis. The function of epididymis is maturation and storage of sperm cells. Third one is the vas deferens. Here you can see where my cursor is. This is testis and here is the epididymis. And it starts from here and coming up to the prostate. This is in orange color. This is a cross section of prostate gland, within which it pierces the substance of prostate, and up to here is the vas deferens. And its function is transport of sperm cell, and at the end this vas deferens is somewhat dilated to form ampulla, where these sperm cells get stored. Lastly, there is ejaculatory duct in the genital duct. Here you can see up to this much, where my cursor is, from here to here. This is ejaculatory duct and its function is to transport seminal fluid. Accessory reproductive glands are seminal vesicle, prostate and bulbourethral gland. Seminal vesicle is secrete fructose. Try to be very specific that the mainly seminal vesicle secrete fructose, which constitutes 60% of the seminal fluid, whereas the prostate secrete glycoprotein. These function and these constituents are very important, constitute 25% of the seminal fluid, whereas the bulbourethral gland, it secretes mucus, which constitute 10% of the seminal fluid, and rest 1 to 5% is the sperm cell. And lastly, there is organ of copulation, that is penis. Coming to testis, regarding its covering, from outside inward it is VAV, V-A-V, that is tunica vaginalis, then tunica albuginea, and within the lobule lining the seminiferous tubule there is tunica vasculosa. Okay, here tunica vasculosa. So the outermost covering is tunica vaginalis, which is double layered, and in this blue one, dark blue, is the vaginal cavity, that is in between the parietal layer and the visceral layer of tunica vaginalis. Tunica vaginalis is nothing but the derivative of the parietal peritoneum. Inside the abdominal cavity it is parietal peritoneum. Within the inguinal canal it is processus vaginalis. Whereas inside the scrotum around the testis it forms tunica vaginalis, which covers testis from all around except the posterior aspect. Second layer is in white color. You can see here where my cursor is the tunica albuginea. Albuginea, albus means white. It is white in color, and it is the very important that it is the ultrasonographic evidence of a male baby. This tunica albuginea, which is very glistening, thick, dense. Okay, and lastly tunica vasculosa, which line these lobules. Tunica albuginea, it surrounds from everywhere, even from posterior aspect. But in posterior aspect, it forms a pocket-like structure, like this, where my cursor is going. This pocket, it invaginate within the substance of testis, okay, and form the mediastinum testis, where rete testis lies. These are net-like structure are rete testis, but this cavity is mediastinum testis. Okay, and now this Tunica albuginea sends many septa, which go anteriorly and join with the Tunica albuginea present on the anterior aspect and divide the substance into many lobules. There are 250 to 300 lobules in one testis, and in one lobule there is 3 to 4 seminiferous tubule. So a total 750 to 1000 seminiferous tubule in one testis, in a single testis. Okay, this you can see the slide of testis, histological slide. This round structure, these are the seminiferous tubule. This in white color, this is Tunica albuginea, okay, and Tunica vasculosa, the innermost one, and the outermost is the Tunica vaginalis, outermost in gray color. Okay, now this round structure here you can appreciate more, and this is also histological slide of testis. Here you can see in the periphery there are immature sperm cell. The sperm cell, the main function of testis is what? Is to produce sperm cell and testosterone. In the periphery there are spermatogonia. The stages of development is like first spermatogonia, then spermatocyte, that is primary and secondary, then spermatid and spermatozoa. Spermatozoa are the mature form, and they are small in size, and inside the lumen, within the lumen in the center, and in the periphery they are spermatogonia, which are large in size. And there are two types of spermatogonia, type A and type B. Type A is a stem cell, whereas the type B is precursor cell. A stem cell is what? When one cell, suppose cell A, give rise to cell B and then cell C, as well as A gives its own progeny, that is A. That type of cell is known as a stem cell, which differentiate into new cell as well as maintain its own progeny. So A is, here is the stem cell, whereas the B is the precursor cell, which is differentiated from A and differentiated into C, but do not maintain its own progeny. That is about the spermatogonia. Okay, and in between there are two types of cell, that is a spermatogenic cell and supporting cell. In the spermatogenic cell there is a spermatogonia, which is type A and type B, and the supporting cell is Sertoli cell, okay, which here you can see. This is also the histological slide of testis, and in between the seminiferous tubule there is interstitial cell of Leydig. This is, here it is written, interstitial cell of Leydig, which secretes testosterone. What is the function of testis? It produces sperm cell as well as the testosterone, which is secreted by Leydig cell, which lies in between the seminiferous tubule, not within, in between the seminiferous tubules. And these are the viva question, but I want to show you what is the function of Sertoli cell. Sir, and why it is called Sir? Because it has done MBA. What is everybody call me Sir, because it has done MBA. How? But how? Sertoli cell done MBA, that is M for Mullerian inhibiting factor. It secretes Mullerian inhibiting factor. B for blood testis barrier. It creates blood testis barrier. And A for androgen binding protein. Mullerian inhibiting factor, Mullerian duct. It the uterus is derived from the Mullerian duct. So this factor suppresses the development of uterus in the male. So Mullerian inhibiting factor. Blood testis barrier, it creates a barrier in between the blood and testis so that no autoimmune reaction can occur. Okay, androgen binding protein, that it binds testosterone. These are the specialized function. In addition to the nutrition, giving nutrition and protection to the sperm cells. And the another thing I want to say that what are the development of sperm? What I have told earlier, that it is spermatogonia, spermatocyte, spermatid, and spermatozoa. How you can memorize? Go CTZ. Go, where in every word there is a spermato. So come to this go. Here is the gonia, that is go, and site C, tid, T, and Z. CTZ, that is chemoreceptor trigger zone, which is responsible for vomiting. We don't like vomiting. So go CTZ. How you can memorize the stages of development of a sperm. Now coming to this viva question. If you get the master slide of a testis, then the examiner will ask you, what is this round structure? In histological slide, what is this round structure? You have to tell it is nothing but the seminiferous tubule. What is the function of testis? It produces a sperm cell as well as the testosterone. What is male gamete and what is female gamete? Male gamete is spermatozoa, whereas the female gamete is secondary oocyte. Tell me the stages of development of a spermatozoa and about its arrangement. So you have to tell that go CTZ, that is first spermatogonia, then a spermatocyte, a spermatid, and a spermatozoa. And the spermatogonia lies in the periphery, whereas the spermatozoa at the center. Okay, there are how many lobules and seminiferous tubule in one testis? There are 250 to 300 lobules, and there are 750 to 1000 seminiferous tubule in one testis. Now another question they can ask, that testosterone is secreted by which cell and where does it lie? So it is secreted by interstitial cell of Leydig, which lies in between the seminiferous tubule, outside the seminiferous tubule. What is the function of Sertoli cell? It is, in addition to the nutrition and protection to the sperm cell, it also does MBA. That is the Mullerian, it secretes Mullerian inhibiting factor, create blood testis barrier, and secrete androgen binding protein. Now coming to epididymis. The genital duct. Here you can see where my cursor is. This is epididymis in green color. This is head of the epididymis. This one is body, and this is tail of the epididymis. Regarding the testis, we came to know that there is seminiferous epithelium, and then two type of cells are there. But what is the histological layer of this genital duct? These are MMS. Here you can see the histological layers. It is MMS, that is in both male and female reproductive system. It is from inside outward. It is MMS, that is mucosa, muscle layer, and serosa or Adventitia. So MMS, that is mucosa. Which type of and mucosa, we all know that mucosa is made up of epithelium and lamina propria. And what type of epithelium we will get in the reproductive system, which I will tell you. And in the muscle layer, there is, in the epididymis, here you can see, in the head there is only circular layer. In body there is circular and outer longitudinal. Okay, and below in the tail there is luckily, that is outer longitudinal, circular, and then inner longitudinal. Okay, so these are the muscle layer, and the lastly there is serosa or adventitia. Okay, what is the difference between serosa and adventitia? Serosa is adventitia attached to the posterior abdominal wall. There is it is not suspended within the peritoneal cavity, but by the if it suspended within the peritoneal cavity via the peritoneal fold, then it is known as serosa. So in some books it is written serosa and in some books it is adventitia. So it's better to tell both. And here you can see the ductal system. Now the examiner will ask you what are the ductal system. So here you can see Sushant Singh Rajput. Here is the Sushant Singh Rajput. So SSR-E-EVE, that is the how you can memorize SSR, that is seminiferous tubule. First there is seminiferous tubule which form the straight tubule, and then rete testis. SSR-E-EVE, that is efferent ductules, vas deferens, and ejaculatory duct. Here you can see SSR, that is seminiferous tubule here, then straight tubule, these are the straight tubule. R, that is rete testis, these are the efferent ductules. EVE, the efferent ductules, then EVE, that is and epididymis, then vas deferens, and then ejaculatory duct. This is the histological slide of epididymis. What is the difference between the histological slide of epididymis and testis? Here in the epididymis, here you can see it is full of sperm. There is in the lumen there is no central hole. But in case of testis, here you can see in the testis there is central lumen which is vacant. In the periphery they are forming the sperm, but there is central empty part in every. Here you can see also this is round structures, and this centrally it is empty, because just by the side of the center the spermatozoa are arranged, because in the testis they are produced in the seminal vesicle, in the seminiferous tubule. But here the main function is the maturation and storage. So in the lumen it is full of there is clump of sperm cell, and second thing, they are tightly condensely packed. In the epididymis these tubules are condensely packed, so they are flattened, they are irregular in shape and size, not circular. Here you can see these are irregular in shape. Okay, and the another big thing, great thing, is the lining epithelium. What we will get in the lining epithelium in epididymis. So we will deal with here the pseudo-stratified columnar epithelium. Pseudo-stratified columnar epithelium, which we will get in the male reproductive system. Pseudo means false, stratified means multi-layered, and columnar is tall. Actually it is single layer, but there are two type of cells are there in the tall columnar, and there is one a small cell. This you can see where my cursor is. This is the basal cell. Okay, and the nucleus lies in the wider part of the cell. So it seems that there are two layers of cell, but actually it is single layered. Some cells reaches the lumen and some cells do not. Okay, but how you can memorize? This world is full of pseudo people. So to understand, to identify this type of people, you have to apply CNS. दिमाग लगाना होता है, CNS. So there are three type of pseudostratified columnar epithelium, that is ciliated, non-ciliated, and stereocilia. Ciliated. Cilia just look like hairs. So urti hui julfe. Urti, that is the upper respiratory tract. Upper respiratory tract, this it bears ciliated type of pseudostratified columnar epithelium. Another one is the non-ciliated, nun, Anu and nun, which is a religious woman who devotes her life for other. And she can be someone's patni, someone's sister, and someone's mother. So here is P for patni and P for prostate, S for sister and S for seminal vesicle, and M for mother and M for male urethra. These are sites where the non-ciliated are found. Now coming to stereocilia. Stereo is what? Is the music system. And music, wow, it gives festive environment. Which and one of those festive environment is EVE, that is epididymis, inner ear, and the ductus deferens. So these are the this is about the epithelium. Now coming to viva questions regarding epididymis. What is the lining epithelium of epididymis? What we have read, EVE, stereocilia. So pseudostratified columnar epithelium with stereocilia. Where else you will get this epithelium? So EVE, that is besides epididymis, it is present in the inner ear and ductus deferens. What is the function of stereocilia? Stereocilia is a mechanosensor. It mainly absorbs 90% of the testicular fluid which lies within the duct. It absorbs. So by this process, this absorption process, it creates a fluid current which is responsible for the movement of a sperm cell. Okay, so it is a mechanosensor. It reacts with the pressure of fluid within the canal. What is the difference between cilia and stereocilia? Cilia is motile, whereas the stereocilia is non-motile. Stereocilia is small in size, whereas cilia is long. Cilia has got 9+2 microtubule arrangement, whereas the stereocilia do not. Stereocilia is mechanosensor, whereas the cilia is responsible for the propulsion, for the movement. Okay, so these are the viva questions.